The genetic code is a system used by the ribosome to translate the language used by the RNA into its corresponding sequence of amino acids in the cytoplasm of both prokaryotes and eukaryotes. Scientists knew that in order to create a protein, you needed an mRNA, ribosome, and a tRNA, but they didn't know which codon coded for which amino acid. In 1961, Nirenberg and Matthaei decided to do an experiment to see if they can match the codons with the amino acids. They started with the poly-U mRNA, which is a strand made up of many of the base uracil, and added it to a test tube that contained a cell-free translation system and mixed it with each individual amino acid. If the protein is radioactive, then the labeled amino acid must be in the protein, and the codon codes for that amino acid. From this experiment, the only time they can make a protein was with the amino acid phenylalanine. This meant that the poly-U mRNA codes for the amino acid phenylalanine. The experiment was repeated with poly-C mRNA, and they were able to show that poly-C mRNA was translated into polypeptides made up of the amino acid proline, while the poly-A mRNA coded for lysine. By the end of this experiment, they were still unsure of how many of the bases containing nucleotides were required to create a codon. So in 1964, Nirenberg collaborated with a different scientist named Leader to try to solve this unsolved question. In this experiment, they added short-synthesized mRNA with one known codon in a test tube. The test tube contained ribosomes and tRNAs with all 20 amino acids, with the one they're testing for being radioactive to label it. After mixing them together, they poured the mixture onto a filter paper. The idea was that anything that was radioactive, like the tRNAs paired with the ribosome bounded mRNA, would stay on the filter paper, and everything that wasn't radioactive, like the tRNA not bounded by the ribosome or being translated, would go through the filter paper. After the mixture was poured and washed, they would check the filter paper to see if it was radioactive. The only way the filter paper could be radioactive is if the trinucleotide coded for the radioactive amino acid that was in the tube. The result of this experiment, after being repeated multiple times, was that they were able to match 61 different combinations of codons out of the 64 codons to their amino acid. They also discovered that the cell decoded mRNAs by reading their nucleotides in groups of 3 called codons. Since there are 64 codons and 61 of them are amino acids, the 3 codons left are stop codons that end translation. The experiments done by Nirenberg and his colleagues resulted in the genetic coding dictionary that we now use today.